Hey guys, Miss Zimmerman here. Today we're going to be talking about the structure of the cell membrane. And if you remember from our discussions through the past couple of weeks, cell membranes are going to be found in all types of cells, plants, animals, and prokaryotes. It is extremely important to the functioning of the cell. It acts as a protective barrier, but it also plays a role into what can go in and out of the cell. So if you look, it's a double layer. There's two layers of these things called phospholipids. We also have a couple of other very important structures that are going to be found within the cell membrane. We have proteins, proteins that go all the way through. We have some proteins that just hang out on one side or the other. We're going to have things called carbohydrate chains connected up to proteins, and all of these different things play a different role for the functioning of the cell membrane. So the first one I want to talk about today is the phospholipid. If you remember, this is a type of lipid or a fat. The phospholipid is unique in that it has a special structure. There is a head region, which we call the polar head, and a tail region, which we call a nonpolar. This end is polar because it is attracted to water. So we call that hydrophilic. So this polar head region is hydrophilic, and then these tail regions are hydrophobic or water-fearing. If you look, the two tails look slightly different. One tail is straight and one tail is kinked. If you think back to lipids, the straight one we call saturated, and the kinked one we call an unsaturated fat. The kink is due to this double bond that we find within that fatty acid tail. Okay, so this is the structure of the phospholipid, and the way that they orient themselves is that the head region is going to face water, and the tail region is going to face away from water or try to get away from water. The plasma membrane, we talked about, serves as a protective barrier. It's what we call selectively permeable, so it's going to allow certain things in and out of the cell, and then it's going to inhibit certain things from getting in or certain things from getting out. The plasma membrane or the cell membrane is also responsible for cell-to-cell recognition. So the cells in our body or the cells in a plant can recognize that that cell is one particular kind, or that that cell is part of your body, and if it's not, then our immune system can help get rid of it. And it also provides anchoring sites for our cytoskeleton. And our cytoskeleton acts as a support system within the cell. Allows it for its shape, but it also allows for movement of different materials inside the cell. Provides a binding site for enzymes. It's going to hold cells together, and it allows the cytoplasm or the fluid inside of the cell to stay inside of the cell. All right, another structure of the cell membrane is proteins. Proteins have a whole variety of functions. They're going to help with transport in and out of the cell. They are going to act as enzymes. They can act as signal sites, so we can have a signal outside of the cell, and that protein is going to allow that signal to travel inside of the cell. Those proteins are what is going to hold two cells together. That cell-to-cell recognition is going to be done by proteins, and the proteins are where the extracellular matrix and the cytoskeleton is going to attach. Now, we talked about that there's two different kinds of proteins. Proteins that are peripheral proteins are proteins that are only found on the outside or the inside of the cell. They do not pass all the way through. Those peripheral proteins are going to provide structural stability and shape to the plasma membrane. The other ones, these ones, are integral proteins, so they are proteins that pass all the way through the membrane. So that's really the main difference between the two. Integral proteins have a lot of different functions, whereas peripheral proteins only have several. Another thing we'll find in there is cholesterol. That cholesterol is going to kind of hide itself in between the phospholipid tails, or this hydrophobic region. And that cholesterol is going to act as a, I guess, fluidity molecule. It's going to allow the cell membrane to be superfluid, meaning moving around a lot and changing shape, or it'll allow that cell plasma membrane or cell membrane to be a little bit more rigid, and we won't see as much movement of these phospholipids. Now, why is that important? Sometimes your cell membrane needs to be a little bit more rigid, especially if temperature's changing or depending on cell type. So depending on outside environmental conditions, we can change the number of cholesterol molecules in there to fit the need of that particular cell. All right, so that's the basic structure. I know that was super quick, but that's kind of the basics of what makes up our cell membrane. So I would like you to pause very quickly and write a 2-sentence or 3-sentence summary on the different parts that make up the cell membrane. All right, now that you have written a little summary of the parts of the cell membrane, I want to talk about types of transport. So how do we get things in and out of the cell membrane? Like we said before, it's selectively permeable or semi-permeable. So that is going to allow it for things to pass through easily or not pass through at all. So if we have really small molecules or molecules that are hydrophobic, so water-fearing, they are going to be able to move through the plasma membrane extremely easy. So some examples of that would be oxygen gas, carbon dioxide, and water. Those guys can are small enough that they can just fit in between here and pass through that membrane very easily. Whereas if we have ions, which are molecules that have a charge, or hydrophilic molecules, or proteins or super big molecules, they aren't going to be able to pass through the spaces in between here. They're just too big. Or in the case of ions, their chemical charge won't allow them to. So they have to have a kind of special way to get through the membrane. Now, if we have oxygen or carbon dioxide that can easily pass through the membrane, it's called simple diffusion. We don't need energy, so we call it passive. It's going to happen naturally, and molecules are going to move from an area of a high concentration to an area of a low concentration. So what I mean is they're going to move from an area where we have a lot of oxygen to an area where we have a little bit of oxygen. I always like to think of this as spraying perfume. If I were to spray perfume right next to my face, I'd smell it right away. But if somebody was standing 15 feet away from me, it would take them a little bit longer to smell it, because the concentration of the perfume was so strong, or there was so much near my face, it's going to spread out to fill out the rest of the room, and it would take a little while for those perfume molecules to reach people in the room around me. The reason that this happens is that molecules naturally have kinetic energy, so they naturally have energy to move, and due to those interactions or those movements, we can move from high to low pretty easy. Here's just an example of what would happen if we were to take some dye and put it into water. You start off, they're all really close, and then those dye molecules, or those red molecules, are going to start to move or diffuse throughout the rest of this beaker. And if we would let this sit without stirring it or moving it, eventually over time we would end up with a glass that looked like this, where the dye was evenly distributed all throughout the glass. Osmosis is a special kind of diffusion, and this is just the diffusion of water. So it's when water moves from a high concentration to a low concentration. Why do we call it something different? I don't know. I think it's just to make our lives as scientists a little bit more difficult. But if we would look at this animation, we start off with a ton of water molecules on this side, and then they are just going to pass through that membrane and even them each other out from the inside to the outside. This idea of osmosis becomes extremely important if we're talking about our body. If you've ever had an IV given to you for fluids, it's important to know the chemistry behind this, because if they give you the wrong type of solution, it could cause your cells to burst or shrink and you become dehydrated. So when we're looking at a cell, there are three, ways that or types of solutions we can put them in. If we put them into what we call an isotonic solution, we're not going to see any movement or net movement of molecules, because the concentration of what we're looking at on the inside of the cell is equal to the environment around it. So in this case, the cell has 90% water, and we're putting it into a solution that has 90% water. And since those concentrations are equal, we're not going to see any movement. And we would call this cell at equilibrium because there's no movement in or out. Now, a cell in a hypotonic solution is going to have a little bit of a different scenario. I always like to think of hypo as a hippo, and hippos are fat. So when we put a cell into a hypotonic solution, we are going to see water move into the cell, and that cell is going to end up getting bigger. If we look at the water concentration, this is 90% water, this is 80% water. So we have a higher concentration of water outside, so that water is going to want to move to the inside so that they can equal out. If the water moves in, then the cell gets bigger. Okay, and then the third type of solution is a hypertonic solution. I always like to think of hyper and hyper people, and hyper people tend to be skinny. So in a hypertonic solution, we're going to see water movement out of the cell, and that cell is going to shrink in size. So if we look at our numbers, we have 95% water here, and we have 85% water on the outside. So there's a higher concentration of water inside, so it's going to want to move out to equal it out, and that water movement out is going to cause that cell to shrink. Okay, here's just another view of what's going on. So here is a red blood cell in an isotonic solution. No net movement because the water concentration is equal on the inside and on the outside. Here is a cell in a hypotonic solution. Water moves in, causing the cell to burst, and that is what we call cytolysis. Okay, so when the cell bursts open, it's called cytolysis. And here's a cell in a hypertonic solution. So water is moving out, causing the cell to shrink, and that is what we call plasmolysis. Here's just another view of those in an actual red blood cell. Isotonic solution, it remains, the same shape. Here's one in a hypotonic, and here's one in a hypertonic. This concept of hypertonic, hypotonic, and isotonic solutions, you have to worry about it with IVs, and that's the reason why you can't drink salt water, because salt water is going to cause the water inside of your cells to move out. But it also, we had some issues with water intoxication, so you can actually drink too much water and die from it. There used to be competitions to win a Wii, which was a Nintendo game console, and there was a lady who died from competing in this because she drank so much water that it caused her cells to become to burst open because the water concentration was just too high inside of her body. And that also happened to a fraternity student who was being hazed, and he was forced to drink a whole bunch of water, and he died from water intoxication. Okay, so that is, simple transport, simple diffusion. Sometimes we have to have what we call facility